Welcome back, Science Squad, for the third experiment of the first Science Squad book, Don't Bomb the Test. Now, this was a very important part of the book because this was how Simon was able to figure out which, which substance to pour into the bomb to neutralize the acid and make sure that the bomb wouldn't go off in the book. He had to figure out, okay, which one of these is a base because the letter said there's an acid and I need to use a base to neutralize the acid, so which one is it? So he had to think really quickly and figure out how to make something called an indicator. An indicator is something you can use to determine if a substance is an acid or if a substance is a base, because it will change color, and depending on what color it changes, that tells you something about its pH. And pH is what scientists use to measure how much of an acid, how acidic something is, and how much of a base something is, or whether it's neutral, which means it's not an acid or a base. So we're going to teach you how to do what Simon did. He went to the cafeteria and got some red cabbage soup. We're going to show you here how to make your own red cabbage indicator. So Sarai is here ready with the procedure in the book. Sarai is on page number 96 of the Science Squad book, and she's going to read through the procedure on page 97 of how to make your own red cabbage indicator. So Sarai, go ahead. What's our first step?
Peel off about 3 to 5 big cabbage leaves.
Okay, so I'll hold this and you go ahead and grab that cabbage. So we got some red cabbage from our local grocery store here. And she's taking off 3 to 5 cabbage leaves. And we decided, how many leaves do we want to take?
5.
We're going for 5, all right? We're going for it. So we're going to use 5 red cabbage leaves, okay? Now the question is, what do you do with the rest of the cabbage after you do this? So feel free to make your own cabbage soup, perhaps, like in the book, or look up a recipe for something that involves red cabbage. But you really only need a few of the leaves. So feel free to figure out some recipes of what you want to do with the rest of this cabbage. All right, how many do we have here? We have 1, we have 2, we have 3, and let's go ahead and get a 4th one. It doesn't have to be perfect. If it comes off in pieces, that's fine, because as we're going to take a look in the procedure, we're going to end up chopping this thing up into little bits anyways, because we're trying to make this into a solution. So we actually are going to be grinding up this cabbage as small as possible so that it will mix with the water and make a solution, a purple solution that we're going to use as an indicator. So you think we got it? Looks like a brain. It does. It does look like a brain. It's kind of cool looking. It looks like a brain. So what do we do now that we peeled off the leaves? Soraya, what's our next step here? What are we going to do next?
Put the leaves into a blender that is half full of water.
Okay, so again, here's another part where you're going to need the help of an adult, a grown-up, to make sure you have permission and you're properly supervised for this part, because using a blender, it's a machine, so it can be dangerous. And inside the blender, how it works is it uses these blades that spin to chop the things up very tiny, so this could be very dangerous. So we got to be very careful with the sharp parts. Make sure you're with an adult. So I'll put that over here for now because we don't need that. So it said we're going to fill this half with water. So here's your water. Here we go. And I'm using a certain type of water, by the way, called distilled water, which just means this water doesn't have any minerals or anything mixed in it. So it's just pure water, pure water. If you don't have distilled water, feel free to use your tap water. But most scientists like distilled water because there's really nothing that will interfere with your experiment within this water. There's no impurities or anything in there. So it will be a nice neutral water, perfect for what we're trying to do here. So you can see Soraya going ahead and putting the red cabbage leaves into the blender cup that's half full of water. Now, as you looked in your procedure, if you don't have a blender, it says you can also take the leaves, put them in a pot of water, and you boil the leaves for about 20 minutes. And I have done that before. Actually, the first time I've ever made red cabbage indicator was that way, by boiling the leaves. But I figured with the blender, it's going to be a lot quicker to make this, because with the boiling water part, there's two things I don't like. One, you're using hot water, and that could be dangerous. So make sure if you're going to use that method, you're with an adult. And two, it takes a while. You got to sit there and boil it for 20 minutes while it makes your entire house smell like red cabbage. As you remember from the story, that smell of the red cabbage soup wasn't very appetizing to Simon or James, or probably anyone else in that school, except apparently Dr. Rosen, who loves cabbage soup. But yeah, so that's why we're going with the blender method. So she's got this in, and you can already see the water starting to kind of turn purple already. Oop, one little piece from this in here. So I'm going to take this part because it's kind of dangerous. I don't want any kids to handle this part, the blade part. All right, get it nice and tight so we don't have any, juice flying around all over here. And go ahead and put that on top of the blender. Okay, great. So now we're going to start the blender. We're going to show you what happens as the blender starts and what's going to happen to this, okay? So we'll get a little close-up of that part. Okay, Soraya's here with the blender. She's going to hit start, and we're going to see this. All right, now that we have our red cabbage juice, which honestly looks more purple than red. The cabbage looks more purple to me than red, but they call it red cabbage, so we're going to call it that. Now we have it, we blended it, but there's going to be a lot of little particles, little pieces of red cabbage in there. So how do we get that out? What's the step we need to do now?
Use a strainer to strain the juice into your cup or container without getting any large chunks of cabbage in it.
All right, so we have a strainer here, and we have a large little cup here. So she put the strainer over the cup, and here's your red cabbage juice. Now, let me do the top part just because there is something sharp here. Don't want to risk any injury, so again, have an adult help you with the blender. Okay, here we go. Yeah, the smell. Red cabbage has a very, very particular smell. All right, so she's pouring it, letting it strain. You can hear and see the liquid coming through here. Okay, as we strain that. So this takes a little while. You got to be patient, pour slowly, allow it to drain slowly, and just continue to go through that process. So we're going to go ahead and show you the end result once this is finished, because this takes a few minutes to really get this all strained out, okay? So here we are, straining it. Soraya was touching it, feeling what this pulp feels like. She's making some observations about the color, how it's looking. So if you feel like you have enough to test the solutions you're going to use, that's fine. But I think we're going to go ahead and create another batch of this. We're gonna double the recipe. We're going to go ahead and use 5 more leaves. We're going to do the same process in the procedure. We're going to grind them in the blender and then strain them out, because I'd like to get even more red cabbage juice than what I have here, so we could use it to test a number of different solutions we're going to find around our house here. We're going to do that, and once we have enough red cabbage juice, you can see we have quite a bit here, but I'd like a little bit more, and then we'll show you the different solutions we found and what we're going to test using our red cabbage juice indicator. All right, Science Squad, here's all the different things we found around the Science Squad lab here that we wanted to test. Now, there's a few things I should mention that, again, without an adult, you probably shouldn't be touching or testing. For example, one of the materials we're going to test is bleach, which is a strong chemical. So if you do not have an adult to handle the bleach and pour the bleach, you should not be testing bleach. It is a strong chemical. But since I'm here to assist, we decided we want to test the bleach and see, is this an acid or is this a base? So we got some vinegar, some bleach, we got some dish soap, our baking soda solution, we have some, what is this? Lemon juice. Lemon juice. What we got here? Some lemon, lime, Sprite, some soda here. This is starch, some starch for laundry, some hand sanitizer, and some window cleaner. And again, we prefer that you use things around your house that are safe to handle, unless you have an adult with you, and proper safety equipment here. We have our safety glasses, we have our gloves, our lab coats. We're ready to do this. So, but again, make sure you use safe chemicals. If you use anything strong like cleaners, especially something like bleach, please don't do that unless there's an adult present to make sure you're being very, very safe when you pour these chemicals. So we're going to pour a little bit into each cup to get it ready so that we'll be able to pour. You can see right here, we finished making our red cabbage juice indicator. So before we use that, we're going to just pour each of these into the cups and let you see what they look like before the red cabbage juice indicator is added. All right, Science Squad, we have everything poured in the cup. Now, before you begin, what you need to do, if you've been reading your procedure, is you're going to go ahead now and you're going to talk about and write about what am I testing. You're going to write that in your book. And then you're going to make some predictions. What we call a hypothesis. A hypothesis in science is making a prediction about which of these you think, when you test them, are going to be an acid, which will be a base, and which will be neutral. And you may be wondering, well, how will I know that? Well, don't forget, on the back of your book, and I'll show this to you in the video, but on the back of the book is what's called an indicator chart. And it has the color change, what color it changes to, and what that pH means. And so it will tell you on the back of the book, if it changes red, for instance, then that's a pH of 1, which means it's an acid. In fact, that's a very strong acid. So hopefully nothing you test turns red. But you'll be using, and I'll show you on the video, a close-up of the pH chart on the back of your book, showing you what pH it is, and you can kind of make a prediction about what you think the pHs will be before you test them, okay? So you're going to go ahead and fill out your Science Squad book here. Talk about some reasons why you think they may be that pH. And then fill out your data, okay? So the name of what you tested and what the pH is based, again, on the chart that you're going to use for reference, okay? What color did it change? And what color does it say that pH is? And you'll be able to then create a chart of your substances and the pHs they were, using evidence to talk about, okay, how did I know this was acid or base? So you're going to use the evidence of what you saw and comparing that with the chart of what pH it is. And I even give you some common pHs of some substances here, okay, that you could kind of look at and see, were our test results the same as what other scientists have found the pHs to be for these types of substances? So maybe what you tested is listed here, but don't look at that yet. Go ahead and try the experiment first, but not until you make your hypothesis, not until you make that prediction of what you think each pH may be. Do you think it's an acid or a base? So I'm going to go ahead and let Soraya start to fill out her Science Squad. Start to fill this out, make her hypothesis, and you at home are going to do the same thing. And once you have your hypothesis, your predictions, you'll go ahead and actually test those with your experiment. All right, so let's get to work. Here's our substances all in the cup, ready for our test. We're about to see what happens to each, what color change, okay, what kind of chemical reaction. We're going to see a chemical reaction and a color change that occurs because of the indicator we're about to use. So let's come over here. Sarai is about to pour in some red cabbage juice into the window cleaner. All right, she's going to move on to the hand sanitizer. And she might need a little bit more in that one. And we're going to mix it around too, so she's going to get something to mix it with and mix it around. So using a popsicle stick or something, you want to mix it around, especially something like that. The hand sanitizer is very thick, so mixing it around will help, all right? Okay, great. Next. You may want to mix each one, but make sure you use a different popsicle stick for each. You don't want to contaminate the chemicals with each other. Okay. That's the starch. So we could see the window cleaner is kind of like this dark green color. The hand sanitizer still is kind of like a purple. And so is the starch. Let's see what happens to this lemon lime soda. All right, she's gonna mix. Ooh, that turned pink. Interesting. All right. How about this lemon juice? All right, she's gonna mix it around with a different popsicle stick, not to contaminate. So that turned even a lighter, like a, I don't know, you definitely could see, I would say almost a lighter shade of pink, right? Like a brighter pink, all right? How about the baking soda solution? Whoa, that turned an entirely different color. That was kind of like the window cleaner, right? Like the color it changed is like this greenish color. Yeah, look at now as she mixes more of the baking soda, it's turning, it looks like
turquoise.
Like teal or turquoise. Cool. All right. So as I compare these colors here, we got teal, turquoise, the lemon and the lemon soda turned kind of pinkish. The starch is kind of a dark purple. The hand sanitizer is a little bit lighter purple. And the glass cleaner is like a dark bluish green. Okay, let's go over here now. So we have this kind of bluish green here with the, now we're testing some soap, some dish soap. And again, the thicker ones definitely need to be stirred, and you should use a different popsicle stick each time so you don't contaminate. So interestingly, anything that's used to clean, like the dish soap or the glass cleaner, they're kind of turning this like green or bluish green color, like the baking soda kind of turned like a bluish color, except these are a little bit darker. All right, now we're here at the bleach. Let's see if that does something similar to the other cleaners, because bleach is used to clean too. Whoa, now the bleach actually looked like it turned like a, what is that? What would you say that color is?
A yellow-green.
Like a yellow-green color. Interesting. And now we have the vinegar. And that turned, what color would you say that turned?
A reddish pink.
Reddish pink. Cool. So here are our colors that changed. And so what you'll do next is you're going to go ahead and use your chart to try to figure out the pH of each of these. All right, so you saw the close-up of the color changes we saw when we added the indicator. And just for reference, we include just the regular indicator with nothing in it, so you could compare this color to all the different colors and see how they changed. And also, this is just the pure distilled water. So you can kind of see, what happens if you just pour the red cabbage juice into water, right? It looks exactly the same, which on your color chart on the back of your book indicates that that's completely neutral. Okay, it's not an acid or a base. So water is neutral, especially distilled water. It's neutral, not an acid or base. So what we discovered here, well, Sarai, what did we discover? What did we find out from this experiment?
That the vinegar was the strongest acid and the bleach was the strongest base.
All right, and we saw that by looking at the indicator chart in our book and comparing what we saw with the chart and making a conclusion from the data we got from our experiment. And so we sort of tried to line them up based on the chart in the book accordingly. So we kind of put them in order from, the strongest acid, pH 1, and then kind of going down based on the color change we saw, putting them in order of pH from like 1 all the way to, what was the pH of the bleach?
14.
Which means it's
base.
Very good. Okay, so interestingly enough, most things used for cleaning are what type of chemical?
A base.
A base. And apparently lemony things with lemon or vinegar, as you should already know from other experiments we did with the book, are acids, and everywhere in between. And some things are pretty close to neutral, which makes sense because a lot of these things that you use in your everyday life, you don't want them to be too much of an acid or a base. They should be pretty close to being a neutral, which you see here. And there you have it. So I hope you had a lot of fun doing that. Feel free to test even more things around your house as long as you have permission and it's things that are safe for you to handle. And go ahead and test your predictions. Make those hypotheses, is it acid or base, especially now that you kind of know some of these things used for cleaning, like soap and those types of things, or tend to be more of a base, and things that are kind of like a lemon or vinegar, things like that, are like an acid. Maybe you could kind of make an even more accurate hypothesis, prediction about what's an acid or base when you start testing more chemicals around your house. So I hope you had fun. We had fun. Did you have fun? You sure?
Yeah.
Okay. She looks tired. Yeah. Science is hard work. But there you go. And thanks. And I look forward to doing even more experiments with you and the squad. And thanks for watching. Don't forget to like and subscribe.